I'm Sean Yusko, and today is February 25th, and this video is for the Unit 2 case connections. Regulation of glucose uptake by cells. A patient presented with a large glioblastoma brain tumor with areas of necrosis inside of that tumor, as seen in the MRI here in white. The patient also received a PET scan using fluorescently labeled D-glucose to assess the extent and metabolism of that tumor, and this can be found here on the right in red. From this scan, it is evident that the tumor cells have taken up considerably more glucose, and this is denser here, than the remainder of the surrounding cells in the brain tissue. So I was asked for this case study to determine the difference in glucose uptake from the tumor and the surrounding tissue. So here already, just from the medical imaging alone, you can see that there is a difference. Okay. So, glucose uptake occurs through glucose transporters in the cell called GLUTs. There are 14 or 15 varieties, but here we'll just discuss a few here. They exist in the body throughout, and they're ubiquitous. All cells have them, but they vary slightly in form, but they all share the same function, and that is to transport glucose inside the cell through the membrane. So much like you've learned in any biology course about cell membranes and sodium-potassium pumps, this operates in much the same fashion. In fact, this GLUT operates sodium dependently as well, so you can see how sodium attaches and the conformation allows for glucose to go through and into the cell. The reason this is necessary is that glucose is hydrophilic and the outsides of these membranes are hydrophobic, so it requires the transporter to go through. Alright, so in the liver we have GLUT2, in the brain we have GLUT1 and 3 varieties, and GLUT4 can be found in skeletal muscle as well as the adipose tissue. So here in the case of our tumor, we're talking about GLUTs 1 and 3. Again, these transporters do differ in size, location, and the conformations that they use, but their behaviors are what actually regulate glucose uptake. Here I'm going to discuss an effect that I believe to be the cause of the tumor growth. So from medical imaging, we can deduce that the GLUT transporters of the tumor cells have been altered in that they no longer efficiently regulate or inhibit the glucose transport across the cell membranes. They're just letting it flood on in. So this results in an influx of glucose uptake, which is why that area, the tumor, was so dense. This is advantageous to the tumor because it allows for rapid growth and greater chance of survival against the body's immune system. The cancerous growth and proliferation does require a great deal of energy. So the cells rev up glucose intake such that they effectively alter their metabolism and begin to convert excess glucose to lactate, despite there being oxygen. So this is known as the Warburg effect. And here in this image you can see how normally it would follow the traditional pathway of glycolysis all the way through oxidative phosphorylation to create 36 ATP per cycle. Now, instead what we see here is that it chooses to create lactate, which only produces about 4 ATP, or even as little as 2, just following the route with anaerobic synthesis. So why does this effect take place? What is the deal here when, optimally we'd want 36, so this tumor is choosing to do 2 or 4? Well, apparently, according to the Warburg effect, despite the fact that this traditional means of glycolysis and the citric acid cycle creating more ATP, it takes considerably longer. You can do 10 times the speed of this reaction by choosing to create lactate instead. So somewhere, somehow, in the mechanism, with these GLUTs and within the cell, the mitochondria, it is choosing to to actively pursue this route and do it at a much more rapid rate in order to create more cells and bypass glycolysis. So because of this phenomenon, this Warburg effect, we see a faster metabolism inside tumor cells that outgrows the rate of the surrounding. Okay, so case 4. In this case, we're presented with a previously healthy 2-month-old baby girl, neonate, who's on a 100% breastfeed diet and has had a lot of trouble sleeping for greater than 2 hours. Besides these symptoms, the baby has been tested and gotten blood glucose levels that are low, so hypoglycemic, and also found that a liver edge 4 centimeters below the right costal margin shows hepatomegaly, as well as the fact that we notice blood glucose levels increase after feeding, but they tank afterwards, so they can't be sustained within nominal levels during fasting. And epinephrine is then injected to induce a response, but the response is contrary to what is expected. Instead, we get an increased blood lactate level. So based on these criteria, we are tasked, or I am tasked, with the mission of trying to determine the most likely diagnosis for this patient. So here are the 4 possible causes that I was asked to investigate. And right here on the left in this figure just shows typically what would happen in a perfect world scenario. So in the case of low blood sugar, we would expect glucagon to be released in order to stimulate glucogenolysis and break down glycogen in order to release sugar into the blood. So this is what we're looking for. This is like the kind of pathway that we're searching for within these 4 answers. So hereditary fructose intolerance. This stems from a deficiency in aldose B. So this aldose B enzyme, if you recall, is the one that breaks down fructose 2,6-bisphosphate into the 2 different 3-carbon molecules, DHAP and G3P, which eventually continue on to form their way to pyruvate. So if you don't have that enzyme, then you can't create pyruvate. So that results in fructose being trapped in the liver, which enlarges the liver. It also presents with reduced amounts of sugar in the urine and fasting hypoglycemia. So we are seeing enlarged liver and hypoglycemia, so far this is consistent. Now, while the signs and symptoms appear consistent with the patient, this is still not a likely diagnosis, and the reason comes from the fact that breast milk comes from predominantly galactose that gets converted into lactose and then back. So fructose does not play a role in this mechanism. So if the baby's diet of just breast milk is the only thing that's going into this child, it's not receiving fructose. It's just breaking down galactose at this point. So a fructose intolerance would have little or no bearing here on the situation. Galactosemia, this is the inability to metabolize galactose. So this is more along the lines of what we're thinking, right? So it originates from inherited defects of either galactokinase, 4-epimerase, or uridyl transferase. And it can present with cataracts, liver failure, mental deterioration, and depletion of inorganic phosphate in the liver. So while this is found primarily in breast milk, it's still not likely that galactosemia is the root cause of this child's situation, because the blood glucose levels would not increase if the child was unable to convert galactose into glucose. So I kind of didn't get it totally right on the first one, in the fact that I didn't mention that galactose can still be broken down into glucose, and so therefore another way we know that hereditary fructose intolerance isn't the case is that whatever the child has been given, it is breaking it down, and it is actually putting glucose into the blood. So that would mean, again, that aldose B is not the issue, because that is clearly there. So it crosses off hereditary fructose intolerance. And then back to our galactosemia, it's the same deal, because even in the way that galactose is processed, it would require that this happen and form glucose, because we're finding that the blood sugar levels do go up after breastfeeding, and then they tank later. So this child is in fact able to metabolize galactose. So A and B are both incorrect. C, glycogen storage disease. This is most likely our candidate here, especially considering that we've been able to whittle down and knock out these two. So in this case, there are 10 varieties, or actually 11 varieties, of glycogen storage diseases, almost all of which present with hepatomegaly and accumulation of glycogen in the liver. They almost all also bring hypoglycemia and other symptoms that the patient presents. Each has a different root cause, and further testing and investigation will be needed to determine which one of these diseases the child has. But 3A, 3B, 6, 8, 9, and 10 are all valid candidates. Beyond the signs and symptoms being consistent with the patient's presentation, the crux here is the epinephrine administration. So epinephrine should trigger glycogenolysis via the alpha agonist pathway, as well as lipolysis and beta oxidation of fatty acids, in order to maintain the blood glucose levels in a fasted state. Instead, this patient's blood lactate level rose, indicating an inability to break down or use glycogen. Lastly, we'll address glucose 6-phosphate dehydrogenase deficiency. So this is an enzyme that partakes in the first non-reversible step in the pentose pathway. Deficiency of this results in hemolytic anemia, which is really not pertinent to this situation at all. So C seems to be our answer. And again, here on this last slide here, we see a list of several of the possible types of glycogen storage diseases, their signs, and their treatments. But C is the valid answer here.